Tools of Molecular Genetics, Part 2, Module 1, Lecture 4 in the Genetics component. Common mutation screening. As stated, mutation is change in the DNA which affects the function of the protein it codes for. Mutation screening. For some conditions, common mutations may cause all or a large percentage of cases. An example of this is sickle cell anemia. Everyone has a mutation resulting in a base pair change that the amino acid glutamic acid is changed to valine. Every single person with sickle cell anemia has the same change in their DNA. One way to look at these is something called restriction enzymes. These are enzymes that recognize a specific sequence of DNA that consists between 4 and 6 base pairs. There are over 3,500 restriction enzymes. Most of them are known as palindromes, which means they will recognize the opposite sequence of GAATTC and GAATTC in the opposite direction. Methods to determine gene function. DNA is made to RNA, which is used as a template for the protein. Southern blotting looks at changes in the DNA, and we taught northern blotting looks at changes in RNA, and western blotting looks at changes in the actual protein. So here we have our DNA that opens up, and we make a copy of RNA, which is then spliced and gone out from the nucleus to the cytoplasm and is used as a template to make our protein. So let's first talk about southern blotting. In southern blotting, we break our DNA into small fragments. We separate the area of interest. We then label it with a fluorescent tag. We transfer that onto filter paper, and then we can look at it. In northern blotting, this allows us to determine the abundance of RNA transcripts. In cancer, this can be a very important method because certain cancer cells amplify certain sequences. Western blotting, this is a method that we use to look at the actual protein or the end product. Some disorders can be difficult to determine changes looking at the DNA. While it's now we're much better at determining sequence changing in Duchenne's muscular dystrophy, for a long time, this was the best method to determine if somebody was affected with this. Do they have the end product known as dystrophin? Here is an individual Duchenne's muscular dystrophy, and there's no protein made. Here's a normal individual, and here's somebody with a milder form, which we're going to be talking about in future lectures. But again, Western blotting allows us to actually look at the product, which is the protein. So let's talk about sickle cell anemia. Mutation scanning techniques can be used to detect for common mutations in southern blotting. As stated earlier, in sickle cell anemia, it's one single amino acid change in every single person. The restriction enzyme MST2 does not cut two fragments when the sickle cell mutation is present because the amino acid sequence is different. So when we look at somebody who is not a carrier, we have this sequence is broken into two pieces. When someone is affected, both copies don't break, so there's just a single band. Whereas carriers have one copy that cannot be broken by the MST2, and then a copy that is seen as two copies. So this method can determine if somebody's affected with sickle cell, if there's a carrier of sickle cell, or if they have the normal wild type allele. Cystic fibrosis is another common disorder. However, unlike sickle cell anemia, there are 1,400 changes in the gene CFTR. Some occur only in single families. These include missense mutations, frame shift mutations, splicing, and nonsense. Delta F508 is the most common in the European population. Here we have a list of various mutation genes that have high mutation rates based on ethnic groups. In the Ashkenazi Jewish population for cystic fibrosis, the risk is approximately 1 in 24. However, the detection rate from sequencing for CF mutations is 94%. So if someone has genetic testing, the risk that they have cystic fibrosis or they're a carrier for cystic fibrosis after testing decreases to 1 in 400. In contrast, in the non-Hispanic Caucasians, even though the risk is similar, 1 in 25, only 88% are detected through this method. So after normal testing, the risk is 1 in 200, about half of that of individuals of Ashkenazi Jewish descent. Individuals Hispanic Americans, the detection rate's only 72%, even though the risk is much lower, 1 in 646. Because the detection rate is so low, the a priori, even after testing, the risk is actually higher than the non-Hispanic Caucasians or Ashkenazi Jewish, even though their risk before testing is much greater than the Hispanics. When we look at African Americans, their risk is only 1 in 65. However, only 65% of mutations are detected, so their risk after testing again is still greater than that of the Ashkenazi and non-Hispanics. And then Asian, also, they have the least risk in these populations of carrying one of these mutations, but only less than 50% are identified. Cystic fibrosis and screening. For CF, screening panels are available which can test for the most common mutations. Negative tests, though, depend on a number of factors. Mutation screening benefit. It's quicker, cheaper way to identify common mutations than sequencing the entire gene. For conditions which are caused by a few common mutations, negative mutation screening can result in a significant decreased risk. However, as stated in the earlier slide, it depends on the ethnic background. Mutation screening limitations. Does not tell you about the presence or absence of rare mutations, and if testing is negative, the risk of being affected is not ruled out, just decreased. Epigenetics is the ever-expanding area of genetics that's likely to become more and more important as you move through your medical career. The gene code is not changed, but the gene is transcribed differently. So even though the actual sequences may not be changed, the ability to transcribe that gene may be changed by other factors other than pure genetics. As a result, no protein is produced. So it doesn't really matter if the sequence is normal. If we can't make the product, it doesn't help. Histone modifications can result in histones being very tightly wound so that transcription does not occur. DNA methylation. This is another method for lack of transcription. The OH is added to the 5th position of the cytosine. This results in the methylation or the gene not being transcribed. And there are a number of human disorders that only a paternal or maternal copy of a specific gene are transcribed. Whole genome or exome sequencing. Single nucleotide polymorphism, or SNP, is a term you're probably going to hear quite a bit. A locus with a different base pair present than commonly seen. In some cases, it affects the gene. In other cases, it's in a non-coding region. Humans are 99.9% similar. Small changes in the genome make the differences between one individual or another. Genome and exome sequencing. Genome, the entire set of genetic instructions consisting of the 23 chromosomes found in the nucleus, as well as small chromosomes found in the cell's mitochondria, are sequenced. There are over 3.1 billion DNA bases. Exome testing is just looking at the sequencing portions of the genome. If you look at the genome as an apple, the exome is the sequencing area. It's just a small fragment of that apple. Genome and exome testing. This is used next generation sequencing, identify genetic causes for diseases when a specific syndrome is not known. It's more efficient and affordable when there's a long list of candidate genes, and identifies mutations in cancer cells. And this is being used more and more for cancer therapy. There also can be sequencing of RNA for gene expression studies. Benefit of exome is it reduces the cost while focusing on the most likely areas in the genome. The human genome. The cost for looking at exome has decreased exponentially. In 2000, we completed the sequencing of the human genome. It cost $1 billion. We can now sequence an entire genome for a few $1,000s. And this is likely to become more and more cost-effective and more used over the series of your career. Next generation sequencing is also known as massive parallel sequencing that makes a whole exome and genome sequencing possible. Using capillary electrophoresis, Sanger sequencing in human genomes, as stated, took over 10 years and cost $3 billion. Fluorescent signals used to determine the sequence similarity in Sanger sequencing, but massive parallel in the rapid format. Samples are run in triad, the patient, the mother, and the father. This helps us to determine candidate genes. There is so much data that if there's a small change in the gene that one of the parents has, and it's thought to be inherited dominant and the parents are not affected, then we would assume that that's just a benign variant. As time goes on, we'll probably need less of these trio studies. Basically, what we do is we cut all the DNA in small fragments, and we add these fragments onto a slide. We add our base pairs, and we amplify thousands and thousands of regions at the same time. This allows us to use fluorescence to determine the genes. This is a complicated process, so we're going to show you a video.
This is the most interesting pathologist in the world, and I would like to talk to you about the next generation sequencing. The process begins with genomic DNA, but the DNA is far too long to be sequenced, so it must be broken into smaller pieces. This is done by a sonar pulse, so we will need a sound so unpleasant it can actually shatter DNA. Thank you very much, Justin. Now, the DNA must be split into two strands, that is, denatured. It is then hybridized to a surface, either a bead or a solid plate. Now, I do not wish to favor one platform over another, so instead, we will hybridize to Chuck Norris's beard. It's a spherical Chuck Norris, but still sexy nonetheless. The DNA fragment is then amplified in either an emulsion PCR reaction or an isothermal bridge amplification reaction. Yes, these are very, very big words indeed, but just watch the video. Here is some music for you. now at last we are left with a clonal cluster of DNA. The DNA is then sequenced together, in this case with fluorescent nucleotides. As they are added, they emit light of a certain color, so that you know which nucleotide was added. Now, of course, there are other ways of doing this, such as monitoring current, but you must agree, this is the most pleasing to the eye. Very beautiful.
So finally, our question: a 6-year-old male has calf hypertrophy, difficulty walking upstairs. There's concern he may have Duchenne's muscular dystrophy. Molecular studies used to determine the amount of dystrophin protein is known as southern blotting, northern blotting, or western blotting.